Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, as you know that we have started a series on food microbiology and today we are conducting yet another lecture in the same series. In today's lecture, we will try and understand spoilage of canned foods, in which we will also try to understand chemical and biological spoilage of food. To discuss this topic, we have with us our subject expert, Dr. Pavas Goswami. Dr. Goswami is Assistant Professor in Department of Microbiology in Bhaskaracharya College of Applied Sciences, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome, Sir.
Thank you. Dear viewers, good afternoon. Today I will be talking about spoilage of canned foods. In my lecture, I will be discussing about chemical spoilage of canned foods, what is the appearance of the unopened container if the food is spoiled. Then I will be discussing biological spoilage of canned foods, grouping of canned foods on the basis of acidity, what are the types of biological spoilage of canned foods, that is caused by thermophilic spore-forming bacteria, and in this I will be covering flat sour spoilage, TA spoilage and sulfide spoilage. I will also be talking about biological spoilage of canned foods which is caused by mesophilic microorganisms, and in this we will be covering the spoilages caused by Clostridium species, Bacillus species, non-spore formers, yeasts and molds. Also I will be discussing the spoilage of canned foods with different acidities in my talk. So to start with, what is food spoilage? Food spoilage can be defined as a disagreeable change in food's normal state. Such changes can be detected by smell, taste, touch or sight. Now, spoilage of canned foods may occur due to chemical reasons and biological reasons. Chemical reasons, mostly spoilage takes place due to the reaction of food material with that of the container, while biological spoilage of the canned food is due to microorganisms. First, coming to chemical spoilage of canned foods, hydrogen swell is one of the most important kind of spoilage of canned foods. This swell results from the pressure of hydrogen gas released by the action of acid of food on the iron of the can. Hydrogen swells are favored by increasing acidities of foods, increasing temperatures of storage, imperfections in the tinning and lacquering of interior of the can, and presence of soluble sulfur and phosphorus compounds. Apart from hydrogen swell, other defects are also caused by interaction between the steel base of the can and the contained food. These changes include discoloration of the inside of the can, discoloration of the food, production of off flavors in the food, cloudiness of liquors or syrups, corrosion or perforation of metal, and loss in nutritive value. So, loss in nutritive value of food due to chemical reactions occurring between the material of can and the food is also a defect. Now, if we have a look at the appearance of the unopened container, normally the ends of the can of food are flat. They are termed as flat, which means that they are slightly concave and a partial vacuum exists in the container. Though we call it flat, but it is partially concave with vacuum inside it. There can be distortions in the appearance of can due to increase in the pressure. These distortions are flippers. A flipper can has flat ends, one of which will become convex when the side of the can is struck sharply or the temperature of contents is increased. Another defect is springer. A springer can has both ends of the can bulged, but one or both the ends will remain concave if pushed in, or if swollen end is pushed in and opposite flat end will pop out. Another defect of the unopened container is soft swell. A can with soft swell has both ends bulged with the gas pressure, and it is low enough to permit the ends of the can to be dented by manual pressure. Hard swell occurs. In this swell, ends are too hard to dent by hand. Another defect is a breather. In this, a can with a minute leak is present, and this leak allows air to move in or out, but it may not necessarily allow microorganisms to enter. This is the appearance of can. Here, this one is the can with flat end. You can see it is slightly concave, while this one is the spoiled can having bulging. Now, coming to biological spoilage of canned foods, there are two main causes of the biological spoilage. These include survival of organisms after the administration of the heat treatment. If we are giving a heat treatment to the food present in can, and if that treatment is not adequate, then cells or spores may survive, and if opportunity is there, they will grow and cause spoilage of food. So, vegetative cells survive the mild heat treatments such as pasteurization of milk, and this results in limited increase in shelf life of food, because some cells have survived. When heat treatments at higher temperatures are given, then the killing of vegetative cells takes place, but spores of bacteria may survive at these temperatures, resulting in spoilage. Another reason for biological spoilage of canned foods is leakage of the container after the heat process, permitting the entry of the organism in the can. That is, suppose we have heat treated the can and organisms have been destroyed, but still if post processing contamination takes place and microorganisms can enter the can, then they will lead to the spoilage of food present in the can. Based on the acidity or pH, canned foods are divided into two broad groups. One is low acid foods. Low acid foods have a pH greater than 4.5, and high acid foods have pH less than 4.5. So, these are the two major groups of canned foods based on the acidity. Now, coming to spoilage of canned foods. The spoilage of canned foods can be carried out by microorganisms which may be thermophilic or mesophilic. Thermophilic bacteria or thermophilic microorganisms are those microorganisms which grow at higher temperatures, and mesophilic microorganisms are those organisms which grow at comparatively low temperatures around 37 degrees centigrade. Now, thermophiles cause spoilage of canned foods, and thermophilic spore forming bacteria are involved. Three most important kinds of biological spoilage of commercially canned foods are flat sour spoilage, TA spoilage, thermophilic anaerobic spoilage, and sulfide spoilage. In flat sour spoilage, they can remain flat during souring. This flat can is an indicative of a unspoiled food, that is, external changes are not being made in the can, perhaps due to low gas pressure or no gas at all. So, without opening the can, you cannot detect the flat sour spoilage. It is caused due to development of lactic acid in the food by flat sour bacteria. Flat sour spoilage occurs chiefly in low acid foods such as peas and corn and is caused by bacillus. Flat sour spoilage of acid foods such as tomatoes and tomato juice is caused by Bacillus coagulans, which is a facultative thermophile. Here this point is important. If a thermophile is a facultative thermophile, then it can grow at the temperatures of mesophilic ranges also and cause spoilage. Various species of bacillus are able to produce acid without gas in food, and these may be mesophilic, facultative thermophile or obligate thermophiles. Spores of mesophiles are least heat resistant and are killed in the heat treatment and are rarely concerned with the flat sour spoilage. Spores of thermophiles are more heat resistant and may survive the heat process to cause flat sour spoilage. Obligate thermophiles such as Bacillus stereothermophilus causes spoilage when the food is held at higher temperatures for longer periods of time. Here this is important. Two factors are required. One is the presence of microorganism and another is the availability of conditions for that microorganism to grow and cause spoilage. So, in this case Bacillus stereothermophilus is a thermophile, and if food is held at higher temperature, it will grow because thermophiles grow at high temperatures. So, if it is held at higher temperature, this provides opportunity for Bacillus stereothermophilus to grow and cause spoilage. Otherwise, if the temperature is low, then even if the spores are present, organism is present, it might not be able to grow in the food and cause spoilage. Facultative thermophiles cause spoilage at ordinary temperatures because they can grow at these temperatures. Sources of flat source spoilage bacteria are plant equipment such as blanchers, sugar, starch and soil. Another kind of thermophilic spoilage is TA spoilage. It is caused by Clostridium thermosaccharolyticum. It is a thermophilic anaerobe not producing hydrogen sulfide. That is, this grows, but it do not produce hydrogen sulfide. Thermophilic spore forming anaerobe forms acid and gas in foods, and the gas produced is a mixture of carbon dioxide and hydrogen. This gas results in swelling and eventually bursting of can if the food is held at high temperature. Spoiled food usually include sour or cheesy odor. Sources of TA spoilage bacteria in canned foods are plant equipment, example blanchers, sugar, starch and soil. Next spoilage which we will be discussing is sulfide or sulfur stinker spoilage. This spoilage is caused by Desulfotomaculum nigrificans. Spores of this bacteria are less heat resistant than the flat sour and TA bacteria. This kind of spoilage reflects gross underprocessing of the food, that is, it suggests that the appropriate temperatures or heat treatments might not have been achieved for the treatment of canned food. Desulfotomaculum nigrificans is an obligate thermophile, so poor cooling of the heat processed food or hot storage is required for the spoilage, because under these conditions only bacteria will grow and it will not grow at lower temperatures. Hydrogen sulfide is formed in the food contaminated with desulfotomaculum. In corn, bluish grey liquid is evident in this kind of spoilage. In peas, hydrogen sulfide odor is present without the marked discoloration. Desulfotomaculum nigrificans is detected by the formation of black colonies due to the production of ferrous sulfide on iron sulfide agar. These colonies are formed by incubation at 55 degrees centigrade. Sources of sulfide spoilage bacteria are plant equipment, example blanchers, sugar, starch, soil and manure. Now, these were the spoilages caused by thermophilic spore forming organisms. Now, coming to the biological spoilage of canned foods caused by mesophilic microorganisms. So, spoilage by mesophilic spore forming bacteria include Mesophilic Clostridium species and Mesophilic Bacillus species. Mesophiles also include non-spore-forming bacteria, and spoilage can also be carried out by yeasts and molds. Most spoilage of canned foods that results due to underprocessing is caused by Mesophilic spore-forming bacteria. Underprocessing suggests that proper heat treatment is not given to the food, and there may also be indication of post-processing contamination after the heat treatment. This will also lead to spoilage, because as such these bacteria may not be able to tolerate the high temperatures used for heat treatment. Now, coming to the spoilage by Mesophilic Clostridium species. Species of Clostridium may act on sugar resulting in butyric acid type fermentation with the production of carbon dioxide and hydrogen. This results in the swelling of the can. Saccharolytic Clostridia, that is, which break down sugar, have a comparatively low heat resistance. Spoilage by these anaerobes takes place in the foods which have been processed at 100 degrees centigrade or less. Acid or medium acid foods are spoiled in this manner. Examples of Mesophilic Clostridium species causing saccharolytic spoilage are Clostridium butyricum and Clostridium pasteurianum. Now, coming to spoilage by Mesophilic Clostridium species. Proteolytic or putrefactive anaerobes decompose proteins with the production of malodorous compounds such as hydrogen sulfide, mercaptans, ammonia, indole and scatol. Putrefactive anaerobes also produce carbon dioxide and hydrogen, which leads to the defects and causes bulging of can. Spores of these bacteria are very heat resistant. These putrefactive anaerobes grow best in the low acid foods such as peas, corn, meat, fish and poultry. Examples of putrefactive bacteria include Clostridium sporogenes, Clostridium putrefaciens and Clostridium botulinum. Clostridium botulinum is an important organism and it is a pathogen causing botulism. Growth and toxin production by Clostridium botulinum is due to several factors that influence spore germination. These factors are composition of food, moisture content of food, pH, oxidation reduction potential, salt content and temperature and time of the storage of food. The microorganisms have extrinsic and intrinsic factors which affect their growth in food. So, all these factors may affect the spoilage as well as toxicity of microorganisms growing in the food. Now, coming to spoilage by Mesophilic Bacillus species. Commercially canned foods have been spoiled by Bacillus species when the cans are poorly evacuated. Bacillus polymixa, Bacillus macerans have been reported to cause spoilage of canned peas, asparagus, spinach, peaches and tomatoes. Bacillus subtilis is involved in the spoilage of home canned foods. These bacillus are usually involved in the spoilage due to post processing contamination. As such, they may not be able to tolerate the heat treatment given to them. Apart from spore formers, spoilage of food is also caused by non-spore-forming bacteria. The presence of non-spore-forming bacteria in canned foods indicates either the mild heat treatment or post-processing contamination through a leak. Vegetative cells of thermoduric bacteria are fairly heat resistant and can withstand pasteurization, which in case of milk is carried out at around 72 degrees centigrade. So, several bacteria can survive pasteurization. Examples of these bacteria are Enterococci, Streptococcus thermophilus, Micrococcus, Lactobacillus and Microbacterium. Lactobacillus causes spoilage of underprocessed tomato products, pears and other fruits. Heterofermentative lactobacilli may release carbon dioxide to cause swell. Lactobacillus may be homofermentative or heterofermentative. Homofermentative lactobacillus usually produce only lactic acid as the main source of main output of fermentation, while heterofermentative lactobacilli are known to produce various acids and gas also. Micrococci causes spoilage of meat pastes. Streptococcus faecalis is known to cause spoilage of canned hams, and coliforms are also known to produce gas resulting in the can swell. Non-gas forming bacteria causing spoilage of canned foods include Pseudomonas alcaligenes, Micrococcus, Flavobacterium, Proteus. Sources of contamination of non-spore formers is cooling water. Once the food is heated, it is then again cooled rapidly to provide a temperature shock. If food is heated and it remains at high temperature, then spore formers can cause spoilage. So, rapid cooling of food is required, and these bacteria can act as a contaminants coming from the water used for cooling of the food product after the heat treatment. Now, spoilage by yeasts, this indicates underprocessing or leakage, because usually yeasts are they cannot tolerate heat or higher processing temperatures. So, if spoilage is occurring, this suggests that post heat contamination has taken place. Fermentative yeasts are known to spoil canned fruits, jellies, jams, fruit juices and sweetened condensed milk. Film yeasts grow on the surface of jellied pickled pork and pickles, olives and etc. Molds are also a causative agent for spoilage of home canned foods. They can spoil foods having high sugar concentration. Aspergillus, Penicillium, Citromyces have been reported to grow on jellies and candied fruits at high sugar concentration. Some molds are fairly heat resistant, and mold Bisoclamis fulva is a pectin fermenting mold that causes spoilage of canned foods. It produces heat resistant endospores. The reference for this talk was Frazier, Food Microbiology. Thank you. Now, in continuation with the spoilage of foods, I will now talk about the spoilage of meats, poultry and eggs. In my lecture, I will be talking about sources of microorganisms in meats, invasion of tissues of microorganisms, then growth of microorganisms in meat, types of spoilage of meats, that is spoilage under aerobic conditions or spoilage under anaerobic conditions. Then I will be discussing spoilage of poultry and spoilage of eggs, in which I will be talking about natural mechanism for preventing invasion of eggs, spoilage of fresh eggs and microbial spoilage of eggs. Spoilage of meats may be due to by its own enzymes or by microbial action or oxidization of fat due to chemical reasons. So, these are the three reasons which may lead to the spoilage of meats. What are the sources of microorganisms for contamination of meats? Exterior of the animal, as we know, the animals as well as humans harbor the microflora, which may be their normal microflora. So, microflora is present on the exterior of the animal. Intestinal tract of animal is also having bacteria. Then knives used for slaughtering may also be contaminated with microorganisms. Cloths, air may be there, they may have microorganisms and may lead to contamination of meat. Then workers may also harbor microorganisms. Several people are known to be carriers of pathogenic microorganisms and they may contaminate meat. Carts, boxes and equipment may also be act as source of microorganisms. So, all these sources lead to addition of microorganisms to the meat or contamination of the meat. Invasion of tissues by microorganisms take place upon the death of animal, and the factors that influence the invasion of microorganisms are the load in the gut of the animal. Greater the number of microorganisms, greater is the invasion of the tissues. To decrease the gut load, starvation of animal for 24 hours before slaughtering is recommended. Another factor is the physiological condition of the animal immediately before slaughter. If the animal is feverish or fatigued, then bacteria are more likely to enter the tissue. Bleeding is apt to be incomplete in such conditions. Glycogen is used up in the fatigue and the pH drop, which takes place from 7.2 to 5.7 is not obtained, and this results in increased number of microorganisms. Method of killing and bleeding and the rate of cooling also affects the invasion of tissues by microorganisms. If psychotrophic bacteria are present, they can grow at low temperature and can spoil meat in the low temperature at the storage at low temperature. Now, microorganisms are able to grow in meat. Meat is the ideal culture medium for microorganisms because it is high in moisture, it is rich in nitrogenous foods, have minerals and accessory growth factors for the growth of microorganisms, have fermentable carbohydrate, that is glycogen, and the pH is also favorable for the microbial growth. Now, The factors that influence the growth of microorganisms in meat are the kind and amount of contamination with microorganisms and the spread of these organisms. If initially the microbial load of skin or of meat is more, of gut is more, then there are chances that the contamination of meat will also increase. The physical properties of meat also affect the growth of microorganisms. For example, the amount of exposed surface of flesh has a considerable effect on the rate of spoilage. Grinding meat greatly increases the surface area and releases moisture and also distributes bacteria throughout the meat, and this leads to increased bacterial growth. Then chemical properties of the meat, presence of glycogen, presence of protein and the other factors also affect the growth of microorganisms in meat. Chemical properties of the meat such as moisture content also affect. If meat is very dry, no microbial growth may occur on it. If it is a little moist, then it may allow mold growth. If it is still moisture, it will allow yeast growth, and if the meat is very moist, then this condition is favorable for the growth of bacteria. Food for microorganisms is also plentiful, but low content of fermentable carbohydrate and the high protein content tend to favor the non-fermenting types of microorganisms. Availability of oxygen and temperature of storage is also important for the growth of microorganisms. Available oxygen affects the type of microorganisms. If oxygen concentration is higher, then aerobes will be growing and spoiling the meat, and the storage temperature, if the meat is stored at low temperature and cyclotrophs are there, then these will cause spoilage even at the low storage temperatures. Now coming to the types of spoilage. Spoilage of meat can be divided into two broad categories, that is spoilage under aerobic conditions and spoilage under anaerobic conditions. Spoilage of meats can also be divided on the basis of type of microorganism causing the spoilage. For example, spoilage caused by bacteria or spoilage caused by yeasts or molds. Coming to spoilage under aerobic conditions, surface slime is one of the most important spoilage of meat. It is the growth of microorganisms on the surface of meat resulting in slime formation and foul odor. Surface slime is caused by species of Pseudomonas, Acinetobacter, Moraxella, Alkali-genes, Streptococcus, Leuconostoc, Bacillus and Micrococcus. Some species of Lactobacillus can produce slime. The temperature and availability of moisture influence the kind of organisms causing slime. At chilling temperatures and high moisture, Pseudomonas and Alkali-genes groups is able to grow and cause slime formation. With less moisture, micrococci and yeast growth is encouraged, and if moisture is further less, then mold growth is favored. Another kind of spoilage is changes in the color of meat pigments. The red color of meat is called bloom, and this color may be changed to shades of green, brown or gray as a result of the production of oxidizing compounds such as peroxides or hydrogen sulfide by bacteria. Species of Lactobacillus and Leuconostoc are known to cause greening of sausages. Another spoilage occurring in aerobic conditions is changes in fats. Several bacteria are known to cause lipolysis. They may produce lipases. Lipolytic bacteria cause lipolysis and accelerate the oxidation of fats. Rancidity of fat is caused by lipolytic species of Pseudomonas and Achromobacter or by yeasts. Those organisms which will be producing lipases will be responsible for changes in fats, and these include both bacteria and yeast. Another kind of defect or spoilage of meat is phosphorescence. This phosphorescence is caused by the growth of luminous bacteria on the surface of meat, and an example of this kind of bacterium is Photobacterium. Various surface colors are also caused due to the growth of pigmented bacteria. Pigmented bacteria, when grow on the surface of meat, they impart the color of their pigment to the surface and leads to spoilage. Examples of these kinds of spoilages are red spot, which is caused by Serratia marcescens, blue color caused by the growth of Pseudomonas cincinnati. Then Micrococcus is responsible for causing yellow discoloration of the meat. Flavobacterium also produces yellow pigment and is responsible for causing yellow discoloration. Then another bacterium, Chromobacterium lividum, is produces greenish blue to brownish black spots. Yellow pigmented cocci and rods are known to cause purple stamping ink discoloration of the surface fat. In this case, fat becomes rancid and there is appearance of peroxides, and the yellow color changes to the greenish shade and later becomes purplish to blue. So, the defect on the surface of meat are caused by the pigmented bacteria, and the defect name depends on the type of pigment which is being produced by the bacteria. Off odors and off taste, usually these are called taints, are also produced by the growth of aerobic bacteria on the surface of meat. There may be soring, which is the defect due to the production of volatile acids such as formic, acetic, butyric and propionic acids, or due to the yeast growth. Microorganisms grow on the surface of meat and results in production of acids, and these acids are responsible for the soring. Another defect of meat or spoilage which takes place under aerobic condition is cold storage flavor. So, cold storage flavor is an indefinite term for a stale flavor which a meat may absorb from the cold storage. Another type of defect or spoilage is caused by actinomycetes. This results in the musty or earthy flavor of the meat. Yeasts are also responsible for causing the aerobic spoilage of meat. They cause sliminess while growing on the surface of meat. They also cause lipolysis because several yeasts are known to produce lipases. Their growth on the surface of meat may also result in the production of off odors and off tastes. Discolorations are also produced by the yeasts, and these discolorations will also depend on the type of yeast which is growing and whether that yeast is producing some pigment or not. So, if pigment is produced, then say pink pigment is produced, then that will be a pink discoloration. Yeast may also cause white discolorations or cream discolorations while growing on the surface of the meat. Aerobic growth of molds are also known to cause the following defects. First one is stickiness. Initially, when mold grows on the surface of the meat becomes sticky to touch. So, incipient growth of mold makes the surface sticky to touch, and this results in stickiness of the meat. Another spoilage is whiskers. Formation of whiskers takes place on the surface of meat, and in this case white fuzzy growth occurs on the surface of meat. Normal vegetative growth may take place, but sporulation may not occur as the temperature may not be suitable for the formation of spores. So, this kind of growth is called whiskers, and in this sporulation does not takes place. Examples of molds producing sporulation on producing whiskers on the surface of meat are Thamnidium ketocladioides, Mucor mucido, Mucor racemosus, and Rhizopus. Then when mold grows on the surface of meat, then according to the type of spores or sporulation, it may also produce discolorations. One such discoloration is the production of black spot. So, black spots are produced by the growth of Cladosporium herbarum, and white spots are produced by the growth of molds Sporotrichum carnis and Geotrichum. Aerobic growth of molds also cause following defects. Now, if the mold is growing, then according to the color it is producing, it may produce various patches. Green patches are produced by Penicillium expansum, Penicillium asperulum, and Penicillium oxalicum. Then mold growth may also cause decomposition of fats when the mold is growing and it is producing lipase. So, if lipase is being produced, then fats present on the meat will be decomposed. Growth of molds also give off odors and off tastes in the meat. Now, another kind of spoilage is spoilage under anaerobic condition. A very common spoilage occurring under anaerobic conditions is souring. This term applies to sour odor and sour taste. Souring can result either due to action of meat's own enzymes during aging and ripening. It can also be produced due to anaerobic production of fatty acids or lactic acid by bacterial action. Proteolysis without putrefaction may also cause souring, and this kind of spoilage is caused by facultative or anaerobic bacteria and is called stinking sour fermentation. In this, acid and gas formation accompany the action of the butyrics, that is bacteria which produces acid, butyric acids. Then Clostridium species and the coliforms grow on carbohydrates and produce stinking sour fermentation. Putrefaction is another important spoilage that takes place in anaerobic conditions. It is the anaerobic decomposition of proteins with the production of foul smelling compounds such as hydrogen sulfide, mercaptans, indole, scatol, ammonia and amines. Putrefaction is caused by several bacteria, for example, Clostridium, Pseudomonas, alkali genes and Proteus species. Another important defect is taint, and in meat, bone taint occurs under anaerobic condition. In this, there is souring or putrefaction of meat near the bones, and this is an important defect occurring in anaerobic conditions. Now, we will talk about spoilage of poultry. Bacterial growth takes place on the surface of skin, lining of the body cavity and any cut surfaces, and the decomposition products diffuse slowly in the poultry meat, leading to spoilage. Eviscerated poultry held at 10 degrees centigrade or below is soiled by Pseudomonas, Torulopsis and Rhodotorula. Above 10 degrees centigrade, micrococci are known to predominate and growth of alkali genes and flavobacterium is also observed. The sources of contamination of poultry is again the surface, and high number of bacteria may be present on the surface of the poultry and may lead to the contamination. Iced cut up poultry also develops slime. This defect is caused by the species of Pseudomonas and alkali genes. Now, coming to the spoilage of eggs. In case of eggs, the sources of the contamination of egg shell are fecal matter of hen, water which is used for washing. Handler again is an important source of bacteria or microorganisms, and the packing material may also add to the contaminants. Initially, the egg may be sterile, but once it can be contaminated by these sources of microorganisms. Total number of microorganisms per shell of hen's egg have been reported in a range of 10 raised to power 2 to 10 raised to power 7. So, high number of microorganisms are present on the surface of the shell of the eggs. Several kinds of bacteria have been recovered from shell, and these include Streptococcus, Staphylococcus, Micrococcus, Sarcina, Arthrobacter, Bacillus, Pseudomonas, Achromobacter, alkali genes, Flavobacterium, Cytophaga and Serratia. There are some natural mechanisms present in eggs which may prevent invasion of eggs. These include presence of eggshell and membranes which may act as physical barriers. Albumin is also present, which is inadequate for growth. It is having a high pH of 9 to 10, so it does not support growth of microorganisms. Then there are low levels of simple nitrogenous compounds. Presence of apoprotein is there which binds riboflavin, so that it is not available for the growth of bacteria. Similarly, there is presence of avidin which ties up biotin, so that it is not available for the bacterial growth. Antiproteolytic factors are also present in egg, and these prevent the action of bacterial proteases. Conalbumin is present which act as a chelator and bind iron. Lysozyme is also there which acts on the cell wall of gram positive bacteria. Now coming to spoilage of fresh eggs, there may be defects occurring in fresh eggs. These defects may include cracks, leaks, then loss of bloom or gloss, stained or dirty spots, meat spots, that is blood clots. Now, to cause this spoilage of an undamaged shell, the organism must contaminate the shell, penetrate the pores of the shell to shell membranes, grow through the shell membrane to reach the egg white, and grow in the egg white to reach the yolk. This is the diagram of egg. Now, here you can see shell is present. So, organism should enter through this shell, should pass this egg membrane, should grow in albumin and then in yolk, so that it can contaminate and spoil egg. Several kinds of bacterial spoilage or rots of eggs are known. These include green rots, which are caused by Pseudomonas fluorescens. The bacterium grows at 0 degrees centigrade. Bright green color of egg white develops in this kind of rot. Yolk disintegrates and blend with white to mask the green color. Odor is lacking or is fruity and sweetish. Colorless rots are also caused by Pseudomonas and Acinetobacter. These rots are detected by candling, and odor is scarcely detectable in these. Black rots are caused by Proteus species and Pseudomonas. In these rots, yolk become blackened and break down the egg, and the whole egg contents have a muddy brown color. Odor is putrid with hydrogen sulfide. Another rot is pink rot, which is caused by Pseudomonas, and in this pinkish precipitate is formed. Similarly, red rot is caused by Serratia because it produces red color. Secondary invaders are also present which cause spoilage of eggs. Fungal spoilage of eggs include spoilage caused by Penicillium cladosporium and Sporotrichum. Pin spot colonies are formed in this kind of spoilage. Superficial fungal spoilage also takes place, and it is supported by formation of whiskers and then luxuriant growth on the surface of egg, and this leads to spoilage of egg. Colored rots of fungi are also caused, such as fungal red rot caused by Sporotrichum and fungal black rot as caused by Cladosporium. Further, off flavors may also be produced by bacteria and fungi, such as musty flavors, earthy flavors, hay odor, fishy flavors and cold storage taste. Thank you.
Dear friends, on that note, we would like to thank Dr. Goswami for coming to our show and delivering this wonderful lecture. And thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.